DNA replication is a process in which we can take our existing genomes or existing set of DNA wrapped up in chromosomes and replicate it or copy it to make another copy. So why do we need more DNA and how do we get more DNA? So we need more DNA because first of all, we have to grow. So we all start out as a single fertilized cell, and then from there you go through embryonic development, fetal development, childhood development, until as an adult you're made up of trillions of cells. So we need more DNA because each new cell is going to get a complete copy of your DNA. Additionally, when you injure yourself, say you cut your skin, the new tissue that fills in the area has to be able to make new DNA so it can make new cells. And how do we get more DNA? That's how we go through the DNA or the process of DNA replication, which we're going to talk about on the next slides. So first of all, the two strands of DNA are complementary, meaning that if you know what one strand is, you can figure out what the other strand is. Because we know that A always pairs with T and G always pairs with C. So remember our GCAT mnemonic, G and C always pair together and A and T always pair together. So what happens in DNA replication is that the two sides of the DNA get separated from each other, and then each original strand becomes one of the strands in a new set of DNA, and then new nucleotides are added to make up the second strand. So we can see that each new strand of DNA contains one side that's made up of the old DNA and one side made up of the new DNA. So we call this semi-conservative replication because we are conserving part of the DNA that we started out with because it's becoming half of the new DNA. So each strand serves as a template for the new strand that's going to be made. Now, DNA replication, like most or if not all chemical reactions in the human body, require enzymes in order to occur. So some of the enzymes involved or required for DNA replication in eukaryotes includes DNA polymerase, helicase, primase, exonuclease, and ligase. And the nice thing about enzymes is, first of all, you notice that they all end in the letters ASE. So that's a good hint that these are enzymes. And the names of the enzymes also kind of tell you what they do. So for example, DNA polymerase is involved in making a polymer of DNA. So it's the one that adds nucleotides, which are the monomers, to the existing strand in order to form a new polymer. Helicase is an enzyme that kind of unwinds the helicase or the helix of DNA. Primase adds a primer to one strand of DNA. Exonuclease goes in and actually cuts out some nucleotides. And ligase helps to kind of glue new nucleotides together. So we'll take a look at the roles of these enzymes on the next slides as well. All right, so first of all, let's take a look at our strand of DNA. So this is our double helix, so our double strand of DNA. So we've got our backbone here, which has the deoxyribose and phosphate, and then we've got the rungs of the ladder. These are going to be our bases here. So this would be where we would see the adenine, thymine, guanine, and cytosine. And remember that the carbons in the deoxyribose are numbered, and notice how the two strands are running in opposite directions. That's going to come into play. That's going to be important later. So the first thing that happens is that the existing strand of DNA has to be unzipped or opened up. So this is called a replication fork because remember, each original strand of DNA is going to become one of the strands in the new DNA, and we're just going to add new nucleotides to either strand to make the second copy. So this strand is going to get a new set of DNA added, and this strand is going to get a new set of DNA added. So the first thing that happens is that helicase protein comes in, and helicase is the one that unwinds the DNA strands. So once again, it's taking the helix and it's making it no longer a helix. Binding proteins then come in order to help hold the replication fork open. Without the binding proteins, the kind of the natural chemical structure of the DNA would want it to kind of twist back into that double helix formation. Another enzyme comes along called primase. And primase is the enzyme that makes a short segment of RNA that's complementary to the DNA. And this RNA is a primer, so it's going to be there kind of temporarily. It just helps to get things started. And then down the line, we're going to cut it out and add DNA nucleotides there instead of RNA. Now, once again, you'll notice that we're going in opposite directions. So the DNA on the top strand here is going from 5' to 3'. And so the RNA also has a direction just like DNA does. So the complementary one is anti-parallel, meaning it goes from 5' to 3' in this direction. Same thing with the bottom row, but it's going in opposite directions. Now, one thing that I want to point out is that, remember, we've got 3 billion base pairs. So if there was only one replication fork, that would take a really, really long time to go through and actually make a complete copy of those 3 billion base pairs. But there are actually multiple replication bubbles that are happening. So each one of these is a replication fork that's getting those primers and starting to do the process of DNA replication. And then once they meet in the middle, now you can see how we end up with 2 separate strands. All right, so coming back to our model here. So we've got our primers here. And then DNA polymerase, which is this purple enzyme here, comes in and it starts adding DNA nucleotides to the primer. Now you'll notice that it can only add the DNA nucleotides to the 3' end of the primer. So you'll see on this top one here, those DNA nucleotides are being added here, whereas down here they're being added on the opposite direction. So you can see it keeps adding and they're going in opposite directions. And as it goes along, that DNA polymerase also kind of proofreads bases. And we'll talk about this a little bit later, but that proofreading helps to prevent mutations from happening. So we keep going in and adding our nucleotides because remember, we want to end up with a completely new strand of DNA. So we'll have 2 strands. But notice how this strand on the top is going to the left. The one on the right is being made going to the right. So this top strand is called the leading strand. And the leading strand is kind of easy because once you get the primer, the polymerase just keeps adding DNA nucleotides, and that just keeps happening all the way down along the length of the DNA. But notice you've got your helicase here, which is unwinding this helix and opening up those bases, and it's moving in this direction. So we've got a problem here because this bottom strand is going in the wrong direction. And we call this the lagging strand. And what the lagging strand does is it kind of plays leapfrog, where there's the RNA primer here in green, and then the DNA nucleotides get added going in the opposite direction of the leading strand. But instead of continuing in this direction, there's another RNA primer added back here, and the DNA nucleotides get added on this side. And this is going to keep happening along the way. So the lagging strand is made discontinuously, so it's not made straight and easy like the leading strand. It actually has to do this kind of leapfrog on the bottom strand, or in this case the lagging strand. And we call these fragments Okazaki fragments, probably because some guy named Okazaki discovered them. All right, so this keeps happening all along the strand of DNA. So you notice how we've got the leading strand is being copied continuously, whereas the lagging strand is being made in those Okazaki fragments. All right, now we've got some RNA that is stuck in our DNA, and that can't be there permanently. So the RNA primers, which are the green areas, have to be removed. So another enzyme comes in called exonuclease, and it comes in and it cuts out those RNA primers. All right. And then another enzyme, ligase, comes in and fills the gap with DNA nucleotides. So it's going to replace the RNA nucleotides with DNA nucleotides, and it's going to help to fix or kind of like stick those nucleotides in order to kind of complete that backbone there. And on the lagging strand, that has to happen multiple times. So on the lagging strand, the bottom one here, that's going to happen here and here. So anywhere where there's a DNA primer, it's going to be removed and then filled in with the proper DNA nucleotide.